I will be presenting on the study I've been conducting with Dr. Ramirez investigating the role of the hippocampal formation in spatial working memory. Our study is looking into the function of the major structures that project to the hippocampus. Two main sources of input are the septal complex, which projects to the hippocampus through the septodentate pathway, and the entorhinal cortex, which projects to the hippocampus ipsilaterally through the perforant pathway and contralaterally through the cross-temporodentate pathway. The hippocampus is considered a critical structure for learning and memory, but other studies have suggested that its afferents also have an important functional role. Last year, Katie Barlos examined the effect of the disconnection surgery on rats' performance in the radial arm maze as an assessment of spatial working memory over the course of 6 weeks, and found that when animals received this procedure, they made a greater number of errors in the radial arm maze for the first 2 weeks of testing and then recovered over time. This disconnection surgery has two main elements: a unilateral transection of the fimbriae fornix and dorsal saltarium, and a contralateral lesion of the entorhinal cortex. This procedure eliminates input from the entorhinal cortex in the right hemisphere and input from the septal complex in the left hemisphere. Our present study investigated whether we would see the same effect of the disconnection surgery on spatial working memory if we used a Y maze instead of a radial arm maze as the behavioral assessment. The behavioral test we use in the Y maze is known as an alternation task, and this assesses an animal's ability to remember which arm of the maze it entered on a previous trial and to choose the opposite arm after a delay. We hypothesize that animals that undergo a disconnection surgery will make a greater number of errors in the Y maze compared to animals that undergo a sham craniotomy. This is the timeline for the behavioral component of the experiment. In the first week, we gentled rats and placed them on a food-restricted diet. We then trained the animals with a series of 10 random forced alternations so they would be accustomed to the maze. Then during the surgery week, I randomly assigned animals to two groups, a group that received the disconnection surgery and a group that received the craniotomy as a control procedure. Following a recovery period, we began 4 weeks of postoperative testing in the Y maze and recorded the number of both total and preservative errors or repeated errors in each session. Once the behavioral testing was done, I completed histological analyses. For the first part of this process, I used a cressyl violet acetate stain to identify the placement of the transection and entorhinal cortex lesion. Then for the second part of this process, I quantified the density of acetylcholinesterase in the dentate gyrus as a measure of cholinergic input from the septodentate pathway. These are some examples of NAICS stain lesion cases. The arrow points to the two layers of interest, the supragranular zone and the outer molecular layer. The top left image shows an example of deafferentation in the left hemisphere, where there's clearly a low level of acetylcholinesterase. The bottom right image shows an example of sprouting response in the right hemisphere, marked by an increase in acetylcholinesterase in the outer molecular layer, which occurs as a result of the loss of input from the entorhinal cortex. Overall, we found that there was significantly lower density of acetylcholinesterase in the left dentate gyrus of lesion cases, marking a significant loss of input from the septodentate pathway, and a greater density of acetylcholinesterase in the outer molecular layer, but not the supragranular zone of the right dentate gyrus of the lesion cases, marking a loss of input from the entorhinal cortex and a sprouting response in the septodentate pathway. The behavioral data shows that rats that received the disconnection surgery did make a significantly greater number of both total and preservative errors in the Y maze over the 4 weeks of testing. The figure on the left shows the average total number of errors, and the right shows the total preservative errors. There's also a significant main effect of time for both types of errors, meaning both groups are improving over the 4 weeks, but there's no evidence of recovery of the lesion group compared to the sham group. Here I have just a summary of our overall findings. Our data is consistent with the hypothesis that the lesion group would perform worse compared to the sham group, but there is no evidence of recovery as we saw in the radial arm maze. These findings suggest that the flow of information through the set of structures is important for spatial working memory, and that the demands of the Y maze are different than the radial arm maze. Going forward, it'd be useful to replicate the study with the full 6 weeks of testing to see if we see recovery later on, to increase the sample size, and to run a study with more selective lesions. And that is all for my presentation. Thank you for taking the time to listen, and thank you to everyone who helped out with the project.